Welcome everyone to the weekly seminar. It's a pleasure for me to introduce my very good friend. Yes, Dr. Guimau de Gorgorió. We studied physics together at the University of Barcelona. Then he traveled for a while, and after he did the master in complex systems in Universitat Autònoma de Barcelona. His master thesis was with Ricard Soler from Universitat Pompeu Fabra, working on the evolution and ecological aspects of cancer systems through mathematical modeling. And this initiated his PhD thesis on the same topic, on this group. He finishes the thesis in 2021, and now he got a postdoc position in the group of Sonia Kefi in the Institut de Sciences de l'Evolution de Montpellier in France. And he's also a member of the Institute of Theory and Natura, of Pirineus. It's independent research collective in a very nice place. And today he will talk about how these ideas and tools from ecology and evolution can help us to understand the interplay between cancer and the immune system. So whenever you want, please.
Yeah, cool. Yeah, thanks for the introduction, Luis. And yeah, thanks for inviting me. It's really nice to be here with old friends and yeah, getting to know Dave Fisk. Yeah, the idea is I was asked to not give a very dense talk, so I will try to, should be an overview, and maybe some parts I will go a bit fast over it, and then we can maybe discuss afterwards. And yeah, I'm currently in Montpellier, but I put my previous research place because this is research that I did mostly during my PhD in Barcelona with Ricard Soler. And yeah, we'll be talking about cancer mostly, and to have a very broad overview. The main idea is that cancer is a disease of abnormal cell replication, uncontrolled cell replication. And without wanting to go too deep, it's characterized by a set of hallmark capacities of processes related to the capacity of grow indefinitely, induce angiogenesis, avoid cell death, and all these capacities. Very broadly, it's the second cause of death worldwide. It's one of the main causes of death worldwide. And maybe despite this feeling of cancer being a more modern disease, is something that we know about since the Egyptians. And since then, we could say that we've been trying to find a cure for the disease. And there has been many incredible advances. There are some cancer types that are mostly curable, but on a general picture, we could say that we are not able to cure cancer. And among all these possible treatments that we have nowadays and that have been developed, we're going to be talking slightly about immunotherapies and the question of why we are really not able, why there are more cancers nowadays and why we're not able to cure them. We could say it's related to the fact that there's more people in the world, so there's obviously more cancer deaths. The age expectancy is increasing, but it's also an issue with drug resistance. There are many tumors, and this is a common sad picture that apparently they are cured by an initial treatment, but eventually they relapse and come back. And one, just to have a basic idea, one of the main reasons for this comeback is heterogeneity, which is the fact that inside a single tumor, we have many different cell types with different phenotypes, doing different things, interacting with one another. So eventually it could be that inside many tumors, there are already cells that will be resistant to the different things that we will try to do to that tumor. And on top of this idea of cancer heterogeneity, it's important to understand that cancer is also a process of ecology, evolution, and development. And Again, in a very broad sense, cancers can be seen as ecosystems. There are many different cell types doing different things, interacting with one another. There's an evolutionary process, of course. Cancers accumulate mutations that allow the cells to acquire different phenotypes to overcome selective barriers of the host. And there's a developmental process, because in the normal picture, we have cells differentiating from stem cells to all the way to a given cell type, like neurons, for example. And cancer cells are able to deviate, to re-navigate these differentiation pathways. And what I think is that the interplay between these three processes is what makes cancer such a difficult disease to understand and cure. And it also makes cancer a complex adaptive system. It's built by many agents, and it involves many layers of complexity, since mutations in single nucleotides of our DNA, all the way to reassembles of chromosomes, interactions between different cell types. So it's really a multi-layer process. And the scale that we're going to be talking about today is interaction between the cell-cell interaction between cancer cells and immune cells. And the idea here is that probably immunotherapy nowadays or immunotherapies are, because there are a large set of those, one of the most relevant or most promising treatment strategy for cancer. And we're going to try to see whether ecology, evolution, and the mathematical description of ecological and evolutionary processes can allow us to understand what are the kind of things that might be happening here. Yeah, to get also a little bit of a glimpse, when we say immunotherapy or immunotherapies, this involves a massive amount of well, not massive, but yeah, up to hundreds of different treatment strategies. So it's not like one therapy. It can involve injecting immune system cells. It can involve molecular. So there's a lot of things going on. But the main idea is to use the immune system to attack the tumor, right? And this can be done through many pathways that we're not going to talk about today. We can discuss afterwards. And the historical process of that, very recently, the Nobel Prize in Medicine was given to people working in immunotherapy, and there's a big hype. Maybe hype's not the word. There's a big trend in using immunotherapy nowadays. It's something that was already there since the end of the 19th century, because William Coley and other people at the time, they were injecting bacteria into tumors, and they were realizing that somehow this was activating the immune system to kill the tumor. But the process by which this was happening was not understood, but it was clear that the immune system could somehow play a role in attacking tumors. But how was really a big question. And yeah, the main thing was that the immune system by itself is such a complex system. It's involving so many different cell types that can do so many different things that it has been, we have needed decades to start understanding what's the things that can be happening in this process. And among all the crazy amount of things that can happen, we're going to be talking about the adaptive immune system, which is those cell types, those group of cells that have the capacity to recognize things, that have the capacity to memorize external previous infections. And you will see more or less how this goes. And the reason why we do that is because these cell types, these T cells that we're going to talk about, these lymphocytes, are those that are able to somehow recognize and attack cancer cells. So are the main agents that we have to consider in immunotherapies. And we're going to see whether these can be understood as an ecological process. And to frame this a little bit with math, I'm just going to overview the mathematical ideas, but we can, of course, discuss that because it's also my background. What people in the 90s started to understand is that we could maybe describe the interaction between cancer cells, and this E stands for effector cells, which is, let's say, the group of immune cells that can attack the tumor. We can understand this interaction in terms of a combination of predation, which means immune cells are able to predate cancer cells, are able to eat somehow cancer cells, together with competition. Both cell types, through secretion of generation of acidic environments, can compete with one another. And this was something that started to go in 1994. But through the combination of these ideas and the fact that they were able to test this with experiments, several interesting things started to appear. And for example, one relevant issue was the fact that this very simple model is able to predict, this is the very simple model, sorry, apart from the, let's say, cancer growths and cancer is eliminated by the immune system, has an intermediate attractor, right? As a stable state in which the cancer is controlled. You have a small tumor controlled by the immune system, but not totally eradicated, right? And this prediction was able to allow us to understand back in the 90s the fact that in some tumors were not controlled by the immune system and others remained very small and controlled by the immune system. And what they realized is that one of the main parameters separating these two states, generating this transition, was immune predation, the capacity of immune cells to recognize the tumor, attach to the cancer cell and kill it, right? Same with what we know from Lotka-Volterra kind of systems. But the problem is that how this predation parameter goes is something that was not understood, or not totally understood, at least in the 90s. And this brings us to what is known as the self versus non-self problem, which is a major issue in the immune system, and is also what's behind autoimmune diseases, which is through a complex set of processes, immune cells are trained to detect and recognize the things that are not from our own bodies. And of course, when this process goes wrong is where autoimmune diseases, for example, start, because immune cells might be attacking things that are from our own body. Here we have an image of an immune cell engulfing a bacteria. This is something, okay, the immune cell recognizes this is an external pathogen, I have to kill it. And this is also a process that we know is super relevant to blood transfusions, in the sense that different people might have blood cells with different antigens in their surface, different proteins that the immune system can recognize as self or non-self. And this is why sometimes if you're transfused with blood from another patient that is not your blood type, the immune system can say, hey, I don't recognize these surface proteins. I don't recognize these markers. I have to attack these cells because they are not from my own body. And the issue is, okay, so the problem with cancer cells is that they are our own cells. They are not external, so why would the immune system recognize these cells and why would it attack it? How can we understand that raw parameter? And the point here is what is known as genome instability, which very broadly can be put as the genomes of cancer cells are completely pervaded by many types of mutations. Okay, so cancer cells have, at the DNA level, have hundreds to thousands, might have some cancers, hundreds to thousands of different mutations. Even chromosomes can be completely rearranged, can have 4 copies of one chromosome and none of another. So there's a lot of mutational noise happening in these cells, which is actually what allows them also to acquire mutations and evolve. And by 2012 to 2014, what was started to be realized is that some of these mutations happening in the genome of cancers were generating different surface proteins. So thanks to mutations, cancer cells, they are blood cells, but cancer cells were cancer cells were changing the surface proteins that they had in their membrane. So suddenly what was understood is that these same genome mutations that the cancers were taking advantage of to evolve, to change their phenotype, was generating surface proteins that we're allowing the immune system to say, hey, this is not a normal cell. This is a cell that has been undergoing mutations. I have to attack this. And this probably too fast introduction brings us to what, like the short questions I will talk about that are most related to this process between mutations and immune recognitions. And on the first hand, I'm going to try to give a very short overview on what's the outcome of this process, right? Okay, so cancers can adapt through mutations, they can evolve, but these same mutations will be used by the immune system to kill the tumor. And what would be the outcomes of this possible trade-off? Who's going to win eventually? Then I'm going to talk a little bit about heterogeneity. What happens when a tumor has many different of these surface proteins, not just one. And then I'm just going to pose a question on an evolutionary process that I don't know how to solve, but I think it might be one of the key steps in cancer immunotherapies. So as I said, first for the first time, the question was, the realization was, it seems that the more mutations that a tumor has, the better that the immune system is at attacking this tumor. And this was quite a thing, because until then, more mutation implied more evolution, a more aggressive kind of tumor. And suddenly mutations could also be a positive thing for the patient. And what we were trying to understand at the time was, what happens if the previous very simple ecological model, what happens if we introduce this trade-off, this fact that mutations can alter the ecological dynamics involved? And in a very simple way, we said, okay, the replication of cancer cells can increase with mutations, provided that nothing necessary for the genome is broken. And maybe we can discuss the details of each aspect if you want afterwards. But the idea is, the more mutations that the tumor has, the better it might be at replicating, provided it's not too much. And on the other hand, what we found was that modeling the capacity of the immune system to recognize these mutations involved too many scales, but there was data available, so that we were able to understand the more mutations that a tumor has, how higher will be the recognition by the immune system. And so in a very not a complex way, in a simple way, this changes slightly the ecological process, and suddenly both cancer replication, immune recognition depend on a single parameter. And what we found is that there is two transitions that are very similar to one another, as we will see, that might be consistent to what we are seeing in experiments and the clinic, which is for low mutation rates, for low instability, the system can only sit at this large tumor, small immune system kind of attractor. But as we start increasing the mutation rate because of this accumulation of antigens, of surface proteins, what we see is, first, there's the appearance of a small cancer, large immune attractor from here, this new attractor here, and even at higher mutation rates, the large cancer attractor is no longer a possibility. So what we're seeing is that it might be that mutation rates are actually displacing the phase space of the system. And interestingly, I wrote down the specific values because these values were in the same range where people in the labs were seeing. The rate at which, by increasing mutation rates of tumor in mice, mostly with using mutagenic therapies, DNA damage, they were seeing that at values similar to these mutation rates, suddenly the immune system could jump into controlling the tumor. So far, this was an initial. Yeah. absolutely. Yeah, true. This is something that should have been written. Yeah, mu are the mutations per gene per, no, the genetic mutations in a gene per cell per division. So it's how often when a cancer cell divides, it will have a mutation in each gene. Okay, and yeah, I have to say I'm balancing between talking more about the maths or the biology. But if mu is a mutation rate, it's you can understand replication as a probability process. And very broadly, if you imagine you have a number of genes that are responsible for replication, which is NR, and you have a number of genes that are called housekeeping, that are genes that you should not break, because if you break those, if you mutate those, the cell will not be able to survive. You end up saying the replication of this cancer cell is its basal replication rate plus the increase related to those replication genes that you have mutated, provided that none of these housekeeping genes have been mutated. So it's also a probabilistic term. So this allows you to relate replication with mutation rates. Yeah. Yeah. Sorry, say that again. Sorry. Yeah. Okay. Yeah. No, exactly. Thanks. The question is right. This is very within the same exact. So let's say that stable human cells have a mutation rate of about, let's say, 10 to the minus 9, something times 10 to the minus 9. And when you are breaking these DNA repair kind of genes, you are jumping from this 10 to the minus 9 all the way to 10 to the minus 5, or even 5 times 10 to the minus 4. So in the end, yeah, it might be, I put 2 values because it's where 2 transitions are happening, but the claim that we have in the paper is more like these transitions from this large region all the way to when this starts to appear or that starts to disappear. We don't have this defined data to know which of the 2, but the general parameter is this 10 to the minus 5, or slightly bigger. That's consistent with the loss of these DNA repair mechanisms. So yeah, it's obvious that this slide is not precise enough with what is happening in terms of data. Yeah. Great. So as I said, the initial picture was for this first, let's 2 to 5 years, there was this strong evidence relating more mutations means more immune response, more immune attack. And what they were starting seeing, this close to 2016, 2018, what they started seeing is that, in fact, it's not only how many mutations, it's not only how unstable the tumor is, but how these mutations are distributed among the tumor, which means if a tumor has, or if all the cells in the tumor have the same antigen, have the same surface proteins, it seems that the immune system will be able to attack this tumor. The immunotherapy will succeed. But on the other hand, in tumors that had many mutations, if these mutations were very distributed, which means you have some cells with this antigen, some other cells with this antigen, and so on, the immune system was failing. So it was started to be seen that the amount of mutations itself was not the best predictor, the best biomarker for how immunotherapy was going to work. And yeah, funny enough, when we were working on this with Ricard Soler, we were also giving this lecture. This was during the COVID year. These are drawings by Ricard, and we were discussing. We're giving lectures in the complex diseases course in Barcelona, and Ricard was talking about antigenic diversity thresholds, which is some very interesting mathematical developments that were done in the beginning of the 90s. And I'm going to have this very small, hopefully, detour to speak about this, and we will see how this might play a role in what I said about the heterogeneity of these antigens. So to frame this, this was something that was done, this research was done in the beginning of the 90s. And for those of you who are not familiar, I recently read that novel, and it has helped me better understand what this meant. It's about the epidemic of AIDS in the 80s in the city of Chicago, and how all the social oppression that was related to that. And one of the main things is that AIDS was not understood, and the relation between HIV and AIDS was not understood in the beginning. And among the many things that were not understood, there's this specific characteristics of the development of AIDS after HIV infection that was really puzzling at the time. And the main idea is AIDS is characterized by HIV infection. You have a very strong response for about 2 to 3 weeks with very high fevers, with a lot of pain. And then for all the way from 6 to 8 years, patients remain healthy. They have very small amounts of viral load. And then after this very long time lapse, suddenly the viral load suddenly explodes and infects the majority of the immune system of these lymphocytes, and AIDS, the auto-acquired immune, the disease is acquired. And the question was obviously, what is happening during this extremely long? How can you have a 3 weeks infection and then a time lapse for about 8 years? Okay, and one of the things they were thinking, okay, maybe the virus is just sleeping somehow, is doing nothing, but they were in clinical trials, they were seeing that actually the virus is really active, and the immune system is also really active at attacking it. And what at 1991, what Martin Novak and Bob May and other colleagues, they proposed was, what if something different is happening? The virus is not latent, but an evolutionary process is happening here. And what the mathematical model they proposed was very simple. I try to simplify it a bit more so that we can discuss it here, but the idea is pretty straightforward, what they were saying. Say, imagine that you have a group of viral strains. It is not a single virus, but all the different mutants of this HIV virus. This is like a population of different strains. And you can imagine that all of them, they replicate at the same rate, and they are killed by those T cells, those immune cells that are able to recognize. And what is important to know here is that T cells can also only have one receptor in the membrane. So if you are a T cell, you will be specialized in recognizing one immune strain, one type of one virus mutant that has one receptor, right? So, but on the other hand, on the opposite process, these T cells are able to grow after recognition of this specific strain, but each T cell will be killed by all the possible viruses. So it is it's obvious, like the kind of thing that might be happening here. The original model was a bit more complex, but if the virus can kill any T cell, but T cells can only kill those viruses that are for which they are trained, it's obvious that there's an imbalance that could happen. And what the maths involved are very simple. We said, okay, so for the immune system to control this type of virus, VI, you need the dynamical equation to be negative, okay? And what they say, okay, but for the whole populations of HIV strains, if this sum is positive, it means that at least one of these terms will be positive, which means that the immune system will, one of the viruses will not be controlled by the immune system. So for this, the whole sum to be negative is a necessary condition for the immune system to control the whole viral infection. And to solve this sum is a very simple thing. This is just n times r, the sum of because each virus can be supposed to have a very similar growth rate. And the sum of all the different T cells, all the different T cell populations is something that is also known, because the total amount of T cells in the body is also pretty much bounded. So suddenly n, which is the number, okay, both sums should have n on top, which is the number of different viral strains and out of different mutants that the HIV population has, suddenly appears in the equation. And what is more important is that for the immune system to control the infection, you need that the number of different strains has to be smaller. That's in respect to something related with the capacity of the immune system to kill them, right? So this brings us to the point in which there is a point in which there are so many different virus mutants that the immune cells will not able to control them. And this is totally translatable to a divide and win kind of process. The immune system is trying to specialize at so many different things that there is a point in which, because of heterogeneity of the viruses, the control is no longer possible. And this is something that was key in understanding what was happening during this very long process. And what was happening is an arms race, an evolutionary race in which the virus is generating more and more and more and more different mutants until it can escape the immune system, right? And this is a story that I also like when people doubt about mathematical modeling of biology, mathematical modeling of biomedicine. This is one very nice success story, and that helped understand, with simple mathematics, that helped understand one very puzzling question. And I think, yeah, we're back from the detour. Remember, we said that in 2016, strong evidence started saying it's not about the amount of antigens, but it's also about how many different antigens they are, how heterogeneous your tumor is. And what we said is, what if this ecological model had something to do with Martin Novak's diversity threshold? And the issue here is that cancer is not a single population. It's actually, like in viruses, a large group of cancer clones that are called. And each cancer clones dies, and this is also a bit quite long. I'm not going to stop too long on this, but it dies depending on how dominant is its antigen, how recognizable, and how good is the immune system at finding this antigen. Okay, the first thing is something we knew from we said before. The more mutations you have, the more probable it is that you generate a very dominant, a very recognizable antigen. And the second thing is what might be prompting this role of heterogeneity. The more rare that a mutation is, if you have a very large tumor, but only some cells have a mutation, the harder it will be for the immune system to find these mutations. And this brings us to a model in which suddenly the death of cancer cells is related to alpha, which is the amount of antigens, and also gamma, which is the frequency of these antigens. Gamma is the frequency of the antigens that this clone I has, which means if a clone carries a lot of antigens, and these antigens are very clonal, are very common across the rest of the tumor, the clone will be killed more easily by the immune system. And again, we can go through that later on, but we pose the same question, which is, what is a minimum condition for the immune system to control the whole group of cancer clones? And what was interesting is that you arrived to the point in which you said what we found is that the average frequency of new antigens in the tumor, which means how homogeneous our tumor is, times not the amount of antigens, but the average amount of antigens per clone. S is the number of different strains. Actually, Novak was using N, is equivalent to the N of the previous model. This has to be larger than a group of parameters related with cancer replication, immune attachment, and so on. And the question is, okay, we knew that only looking at the amount of antigens might not be enough to understand immunotherapy. Yeah.
Can you remind me again what is alpha?
Yeah, alpha is the amount of alpha I is the amount of antigens that this clone, this strain I, has. The amount of imagine a group of cancer cells has 100 mutations, and there are 10 of these 100 mutations that are generating proteins in the surface that the immune system can recognize, are generating these antigens, these neoantigens that the immune system can recognize. So the more of these antigenic mutations that you have, the more probable is for the immune system to recognize you, right? So what I said in like the original view was like, okay, the more mutations, more antigens, more recognition, right? But it was seen that it's not only about that. It's also related. It's not only about how many antigens a tumor is generating, but it's also how many antigens does each clone has on average, and more importantly, whether these antigens are very frequent across the tumor, like all the different cells have the same, or they are like the frequency of these antigens is very slow, which means one cell has one antigen, another group of cells has another group of cells has another, right? And what we found was that this is a very common way to plot things in biomedicine, which is survival curves. You take I don't know if I have to take too long, but it's interesting to understand. You take a group of patients and you take the lowest half in this metric. These are the patients that have less antigens. These are the patients that have more antigens from just by dividing into the groups. And you plot how long they are surviving, after immunotherapy, which means the faster that this decays means the shortest time that they are surviving in immunotherapy. So clearly it's obvious that the blue patients, which are the ones that have low amount of antigens, are doing a bit worse than the ones that have high amount of antigens in terms of survival. And what we found is that if instead of using this metric in the data, we start taking into account the frequency of antigens and this average antigens per clone, the metric that our model was highlighting, we find that this is a much better predictor of who is being, when immunotherapy is succeeding. So suddenly, if we use this to characterize our patients, we see that now it is more clear that the ones with low frequency or low amount of antigens per clone are not surviving after immunotherapy, and the others are doing clearly much better. So thanks to the ideas developed in the 90s, it seems that something similar might be playing a role in cancer. And yeah, this is the two main databases of patients treated with immunotherapy. And the good thing is that now we start having single cell data on antigens. So all these things can be counted. There's data for all of that. And if you don't, well, we can have questions afterwards. I will just finish here with just what is more of an open question. So it's not no more results, but this is something that I believe might be one of the most relevant questions in immunotherapy, like in present day immunotherapy, which is, okay, the previous picture was a very short time scale picture, was an ecological dynamics picture. So you have a tumor, you activate immune attack, is the immune system gonna control the tumor or not? These kind of questions. But what is happening in many cases is that after an initial attack by the immune system, there's a longer phase in which not the whole cancer is eliminated, but there's in fact a process by which it seems that the cancer cells are able to modulate the antigens they have.
You're fine.
Is that a question? Okay. Yeah, in my PhD thesis, I had some really fun interventions as well. Okay, so it seems that through evolutionary process, cancer cells can modulate the number of mutations, the number of antigens that they have. Okay, so it's not only an ecological process, but also an evolutionary one. And the point is that the adaptive immune system, as the name says as well, is an adaptive process. So it can also be able to recognize that the tumor is silencing some mutations so that it's not recognizable. And so there seems to be this parallel arms race, this evolutionary process that might really guide whether the tumor will be able to eventually escape the immune system. And the outcome of this evolutionary process between these two populations, it appears as if there are two well-defined trajectories, which is either the tumor has a lot of mutations, is evolving really rapidly, and is also being attacked by the immune system, or on the other hand, the tumor is mostly stable, not many mutations, very silent, and the immune system is not reacting. And it seems that you either see the two extremes in cancer types. And the possibility here is something that we've been discussing with Jeffrey West, which is a good colleague from Florida. The possibility here would be that there's like evolutionary process, and this is something that, again, we know from ecology, in which we could maybe describe the evolutionary dynamics of these mutational loads between the two populations, and maybe try to understand if these two options, you either just go the mutations way or you go the silent way. If this might be maybe what are known as evolutionary stable strategies in coevolutionary process. And this is an open question for if someone is interested. I'm super curious about this. And yeah, that's all so far. I want to thank, of course, Luis for inviting, and all of you for attending. I'm going to wait. How hard was that? And yeah, well, Ricard and other people with whom I collaborated on this, and I've been working on these years. Yeah.
Okay, thank you, Gim, for this very, very interesting talk. I think we learned quite a lot of things from the field. We have some time for questions here in the audience or online. If there is none, I have a few of them. I think maybe they are too broad. I found very interesting this idea that both the AIDS problem and then this cancer can be seen as somehow as evolutionary race or competition. I wonder that somehow this kind of dynamics might predict that the immune system can eradicate by itself the virus. And I wonder why this never happens. Does it make sense? You know, during, for instance, all this latent period, it could be, no, that for some fluctuations, actually, some patients could actually.
Yeah. Like I'm not an expert in HIV, but it was seen. It was really uncommon, but some patients didn't jump to the HIV phase. But it was really uncommon. Like this was a super common picture, and it's related to the fact that that HIV is not infecting cells in general, but immune cells. So it's actually infecting those cells that would be responsible for eliminating them. So it's a very fine-tuned thing.
Yes.
So, first of all, thank you for the talk. It's beautiful.
Yeah,
I really liked it. So your model focuses only on this particular therapy, immunotherapy, right?
Mm-hmm.
But usually, I mean, a large part of the patients go through several treatments, like chemo, whatever. Would it be possible to include those into the model?
Yeah. I'm sorry, I didn't want to go into that. So for both, like, let's say question 1 and question 2, like the works we did involved combination therapy. For example, I didn't mention it, but in this first model, we say, okay, the capacity of cancer to replicate, the capacity of the immune system to recognize, these depend on mutations. But in the paper, we also mentioned that M, which is the rate of migration of immune cells towards the tumor, is something that is really modulated, can be really modulated by some types of immunotherapies. While on the other hand, the way where you touch mu is something that is not exactly immunotherapy. It's just mutagenic kind of therapy. So it was, and you could also think about adding a death there related to a more like basal chemotherapy, killing cancer cells, or probably killing both. And for this question that I mentioned, if you remember, we're saying, okay, so the immune system will react better to those tumors that are very homogeneous, that look more or less the same. But normally, what you will have is a tumor that has many different cell types. So it's not at all, the immune system would react better if you only had this, let's say, the blue part of the tumor. And what we were proposing there is that, in fact, this heterogeneity can be reduced by using other therapies that imagine that, for example, this blue part of the tumor is resistant to a type of targeted or molecular therapy that you have. The rest of the tumor is sensitive. So if you give this particular therapy, you will eradicate all other cells. You are really able to modulate heterogeneity with using different combinations. But yeah, these are only examples, of course. And yet, as he mentioned, like nowadays, cancer is usually treated in combination because it's pretty well accepted that using just one strategy will not work.
Thank you. Just for curiosity, but when you were modeling cell cancers and immune cells like a predator prey, then it had surprised me that the growth terms for the cancer cells is actually a logistic growth. There you have a carrying capacity, and just for curiosity, but like in the common knowledge, it seems like cancer cells grow exponentially without control, but here you are establishing some carrying capacity. So can you just tell me why?
This is a good, like, I think the field of cancer, like initially in the 80s, the studies that were understanding the shape of this function were really relevant. The first thing, if you could see the growth curve of a tumor, is something that really slows down almost until saturation, and is bounded by the size of the tissue. So it's true that cancer cells can populate tissues in a more dense way than normal cells, so they have the capacity to increase their carrying capacity. So they can, but what is really killing the patients is not the cancer growing indefinitely, but actually moving to other sides of the body, which is 92% of cancer deaths are because of metastasis. So if this is the model of a tumor, it's the model of when we say a tumor is a bulk of cells, a group of cells, and this tumor has a finite amount of carrying capacity. It's not that it will grow indefinitely, and the issue is not exponential growth, but migration to other parts of the body.
Just another curiosity, but I wonder what is like the typical response or reaction of the people from medical doctors, pharmaceutical industry, when you approach them with this kind of mathematical models and its predictive power. And what do you expect that the outcome of this approach would be in the future?
Yeah, for some reason, like here now I'm working in theoretical ecology, and models are extremely accepted. But yeah, mathematical models of cancer are very rare, and it's mostly thanks to these people in Florida. In the Moffitt Cancer Center is like a massive private research institution in the US, you know, like big US kind of thing. And they have like a whole department of mathematical oncology. And thanks to these people, they have all the process from models to clinical trials. So they are using therapy strategies that they have designed from the maths. And thanks to these people, I think like the mathematical oncology is becoming a thing. Well, not only these people, but they have, yeah. Okay. So we're no longer just like playing around with toy models, but it's hard. It's obviously hard. There's like the simplification of these models is crazy. There are like so many layers of molecular biology in every process.
Okay, I think we can close the session with this, and we can thank the speaker again.
Yeah.
Would be great.